Hi everyone, welcome back to the second video. So in this video we're going to talk about chromosomal mutations. There are two major types of chromosomal changes, and that is a change in chromosomal number and a change in chromosomal arrangement. So a change in chromosomal number results from non-disjunction, and that can be called a polyploidy or an aneuploidy. And then a change in chromosomal arrangement can result from insertion, deletion, inversion, or translocation. So non-disjunction means not coming apart. So when the chromosome pairs line up in metaphase and then they're going to pull apart in anaphase 1, or in anaphase 1, the homologous chromosomes are not going to separate in anaphase 1, and then in anaphase 2, the sister chromatids would not separate. So resulting in this would be if the homologous chromosomes do not separate in anaphase 1, then there would be an unequal amount of homologous chromosomes going to this side. So both of them would go over to one side, resulting in both of them being in the cell, and then the other cell wouldn't have any. And then the same thing with the sister chromatids. 3 of them could separate to this side, and then only 1 of them could separate to another cell. So then there would result in imbalance of chromosomes in the cell. For example, afterwards ending up, a sperm cell could end up with 24 chromosomes instead of 23, or it could also end up with 22 chromosomes instead of the original 23 it needs. Same thing with an egg cell, you could have 23 normally, or you could have 24 on accident, or 22 instead. So here is a picture of it from meiosis. So it starts out normally here in meiosis 1. The tetrads are lining up in the middle with those homologous pairs. And then, as you can see, the homologous pair or tetrad does not separate here in this anaphase stage. So both of them are going to go over to this side, leaving this side with none. So as a result, this cell will have no little blue chromosome in here. Instead, it will be over here, and it will have 3 chromosomes over here, and this one will only have 1. So at the end, it would be haploid number +1 for these two, and this one would be haploid number minus one. So these two would have fewer chromosomes than needed, and these two would have more chromosomes than needed in their gametes. So polyploidy means chromosomal number that contains extra copies of every chromosome pair. So that means every chromosome is going to be have an extra one. So R23 would have an extra, R23 plus the extra, so 46, would have an extra 23. An example of this would be triploidy. So the chromosomal number in which there are 3 copies of all autosomes and 3 sex chromosomes. So like I said before, so 46 would result in an extra 23 instead. Here's an example of a karyotype showing you the extra chromosome for each one for a triploid. So as you can see, there's 3 chromosomes for each chromosome number instead of only 2. And this would be a girl because it has 3 X chromosomes and no Y. An aneuploidy is a chromosome number that results in more or less than 46 chromosomes, and it only affects 1 chromosomal pair. So it's not going to be an extra of all 46. It's going to be, say, if you had an extra of chromosome 1, it would just be, you would have an extra of that 1 chromosome. So these 2 examples are monosomy and trisomy. So monosomy would mean you would have a missing chromosome. So, meaning 1, so you would only have 1 chromosome then. And then a trisomy would mean an extra, you have an extra chromosome. So you would have 3 copies instead of just 2. So here in monosomy you have 1, and then in here you will have 3 in trisomy. An example of trisomy is trisomy 21, which we had talked about in class before. So now you know trisomy 21, or Down syndrome, results in non-disjunction of those chromosomes in anaphase. So it involves the presence of an extra copy of chromosome 21. This disorder is the only one that allows survival into adulthood. Other trisomies do not allow individuals to survive after birth, or during birth, or even before birth. They will not survive. So if you look on here, this karyotype, right down here it shows you the 3rd chromosome for chromosome 21. So this is an example of a trisomy. And you can see the chromosome is smaller than the rest of them, so they don't have as many genes on them, but it still can affect the individual in many ways. So now on to change in chromosomal arrangement. So this isn't going to be a whole other chromosome that is going to be added or deleted. It's just going to be genes on the chromosome. So the entire gene is going to be missing, duplicated, or misplaced due to these 4 types of mutations, not just 1 or 2 nucleotides like we discussed in the gene mutations. So a whole gene is made up of thousands of nucleotides. So at this point it's not just going to affect 1 nucleotide, but it's going to affect 1 gene or many genes. So translocations, duplications, inversions, and deletions are all types of mutations that are going to result in chromosomal arrangement change. So first one is going to be duplication. So all of these on here, A, B, C, D, E, F, G, are all genes. So A is a gene, B is a gene, A is made up of thousands of nucleotides, and B is made up of thousands of nucleotides. So over here you can see that D and E are now going to be replicated. So one segment of this chromosome is now replicated, making it longer, and then it could also be replicated again, making there be 3 repeats of D and E. So here there's only 1 segment that's repeated, and here it repeats 1, 2, 3 times after the original one. So you can see how it gets longer than the normal chromosome. That would be a duplication. inversion, or also meaning to flip. So here you have G, H, and I to begin with, and then that's totally going to flip around. So I is going to be first, and then H, and then G, which then changes the chromosome sequence of genes, which then can also affect the protein that is made from that amino acid, and it would all get mixed up then. But remember, not all mutations are bad. So maybe this inversion could have been good, and then it would be passed on in the genes of the offspring of this individual that has this inversion. Because they're not always bad, you never know, it could end up being a good thing. The next one is translocation. So a lot of people get this mixed up with crossing over. Don't think that this is crossing over of like that occurs in meiosis. This is actually going to be occurring on two different chromosomes. So in crossing over, you cross over with two homologous chromosomes, but here you're translocating genes onto non-homologous chromosomes, so they do not belong on the other chromosome. So changing them is bad. So this one up here is starting out very long, and it has H, I, and J on it. And then down here, this one, its non-homologous chromosome, would be having very small amount of genes, and it just has this N on here, it's highlighting. So then if you translocate these genes, this N will go on the longer chromosome, and then that H, I, and J will go on this shorter chromosome, making this originally very long chromosome shorter, and this shorter chromosome a little longer than it originally was. So it involves two interacting chromosomes, two different ones, not the same chromosome pair. And then the last one would be a deletion. So you're totally deleting the gene from the segment on the chromosome. So you can see here, C is on the chromosome, A, B, C, D, E, F, G, so on. And here, the total C is deleted. It just goes A, B, D, sorry, the C is not there, A, B, D, and the chromosome is then shortened. So it loses the genetic material that is needed in that C gene, and then that can account for many genetic disorders if it's totally gone. And that is it. So make sure you know the difference between a gene mutation affecting one nucleotide and between chromosomal mutations affecting either a total adding or missing of chromosome, or a gene added or missing, and so on. All the different examples we went over. Make sure you look over your YGK sheet, and I will see you in class. And thanks for watching my first OB ever.